the Deputy Vice Chancellor Administration, Deputy Vice Chancellor Academic, the Registrar, University Librarian, Provost of the College of Medicine, the Deputy Provost, the Dean of Science, Deans of other faculties, the Postgraduate School, and of students, the Rectors of institutes, heads of departments, distinguished guests, ladies and gentlemen. The 18th in the series of University of Ibadan Inaugural Lectures for the 2015-2016 session will be delivered by Professor Obi Alexander Ugwumba of the Department of Zoology on behalf of the Faculty of Science. The inaugural lecturer of today was born on Monday, 10th of March, 1956, to Mr. Nelson Oti Ugwumba, teacher Oti, who trained at the famous Opodel Institute, Calabar, and Forabe College, Freetown, Sierra Leone, and Lucy Ugokwo Ugwumba of Arochukwu, Abia State. Alex is the sixth of 7 children of his parents. He attended Wilcox Primary School, Aba, from 1961 to 1966, and Ngwa High School, Aba, from January 1967 till December 1971. After his stint as an employee of United Bank for Africa, he attended Baptist Academy, Shepherd Hill, Ikorodu Road, Lagos, for the higher school certificate between 1974 and 1976. Later that year, he was offered admission to study for the degree of Bachelor of Science in Zoology in this university, for which purpose he utilized the federal government scholarship during the 1976-77 and the 1977-78 sessions, and graduated with a second-class honors degree per division in 1979. He participated in the National Youth Service Corps scheme as a science tutor at Sacred Art College, Akure, from August 1979 till July 1980. He registered for a higher degree program of the University of Lagos in October 1980, during which, in joint partnership with the Environmental Protection Agency, Washington, D.C., he obtained certificate in waste management, EPA certificate in 1983, and also utilized the University of Lagos Bursary Award from the 1980-81 session to the 1983-84 session. He completed his PhD degree in zoology in September 1984. The then Dr. Alex Ugwumba joined the services of this university as a lecturer grade 2 in our Department of Zoology with effect from 15th April 1985. He was promoted lecturer grade 1 with effect from 1st October 1989. Senior lecturer with effect from 1st October 1993, and professor with effect from 1st October 1999. Till date, he has to his credit over 100 publications in reputable peer-reviewed journals and has supervised over 120 BSc, 62 MSc dissertations, as well as 10 PhD theses. Within this university, Professor Ugwumba has served meritoriously in several capacities, including, but not limited to, being member of Senate 1998 till date, acting director, Zoological Garden 1990 to 1995, Warden Nnamdi Azikiwe Hall, 1st October 1995 till 30th September 2001, and head of his department from 1st August 2005 till 31st July 2008. Currently, is the chairman of the University Ad Hoc Committee on Accreditation and University Ranking. And even while on sabbatical at Canadian University, he has not failed to attend any of the meetings of this committee over the last 9 months, and we remain very grateful to Professor Ugwumba. Outside this university, Professor Alex Ugwumba has served as chairman, science sub-team, NUC accreditation to Nigerian universities from 2004 till date. Resource person, Commonwealth Training Program for Aquaculture Excursionists in Commonwealth Africa, 1998. Is listed in Poster of Experts, Commonwealth Secretariat for Developing Countries Programs in Aquaculture and Fisheries Development, 1998. Executive Committee Member, Oyo State Fishermen Society of Nigeria, Member, Editorial Board, Nigerian Association of Aquatic Sciences. Fellow, Nigerian Institute of Biology. Fellow, Fisheries Societies of Nigeria. Member, Nigeria Association of Aquatic Sciences. Member, Ecological Society of Nigeria. Member, Network of Tropical Aquatic Scientists. Member, Network of Tropical Fisheries Scientists. Member, Zoological Society of Nigeria. He served as Editor-in-Chief, The Zoologist, 2001 to 2010. Also, first National Vice President, Zoological Society of Nigeria, from 2011 to 2015. And from 2015 till date, is National President of the Zoological Society of Nigeria. He has been an external, Professor Ugwumba has been external examiner and assessor to many Nigerian and West African universities. Professor Ugwumba is highly sociable, amiable, and friendly. He's an active member of our senior staff club, of which I'm a card-carrying member. I'm also a card-carrying member of the senior staff club. And he was our president from November 1998 till November 2000. He is happily married to his head of department, Professor Adia Alda Alex Ugwumba, his best friend and university classmate, and the marriage is blessed with children. It's now my pleasure to call on Professor Obi Alexander Ugwumba to deliver his inaugural lecture titled From Surface Waters to the Deep Sea Bed, Fish, the Real Masters of the Aqua World. Professor Ugwumba.
Chancellor Administration, Deputy Vice Chancellor Academic, Registrar, Librarian, Provost College of Medicine, Dean Faculty of Science, Dean of Postgraduate School, Deans of other faculties and of students, Directors of institutes, distinguished ladies and gentlemen. It is with great honor and humility I stand before you to deliver the 18th of the 2015-2016 inaugural lectures of this great university on behalf of the Faculty of Science. This is a landmark presentation, for I represent the first grandson of our teachers' teachers to present an inaugural from the department. Other inaugural lectures from the department started from 1975 by Professor F.M. Okoli through Professor Toye, Nwagu, Fagade, Okore, and Professor Hassan. My inaugural lecture after 17 years on the professorial chair suggests that inaugural lectures from the Department of Zoology do not come too soon like others, either by the letter Z, which comes after other letters, or by the numerous number of professors in the faculty now. Things have changed so much, and a lot of slots have been allocated to the Faculty of Science. I thank the Dean of Science, Professor Nilide, who used seniority to slot me in this year over younger, vibrant, and other colleagues. To my other colleagues, my prayer is that yours will come at the anointed time, soon, in the name of Almighty God. My lecture will cover the biology of representative fishes from the surface water to the deep sea bed. I'll look at limnology of inland waters vis-a-vis aquatic health, fisheries, and aquatic pollution. I'll look at practical application of both of them in the culture of fish for mankind. I'll have a peep at my contribution from books, manuals, and monographs. I will try and anchor in biodiversity, conservation, and pollution in our marine environment. Zoology. Zoology is the study of animal science with topics ranging in science from molecules studied in the laboratories to animal societies and ecological studies by field observers. It also ranges in time to physiological and reproductive processes taking place less than a second to evolutionary histories extending over millions of years. Zoology is a science close to human concern, and as humans are animals, this closeness often poses problems to human beings because we don't know when to separate humans from animals. All the history of biology started with Aristotle's zoology, one of the earliest studies of the natural world, a true defining moment in scientific history. Aristotle's methods and theories seem a little primitive to modern humanity with his genome codes. However, his work was a quantum leap in the building of human knowledge. Every modern biologist is advised to study Aristotle's zoology as a perfect example of how to build up a store of knowledge based on careful observation. It is this brilliant mind that cemented everybody to call Aristotle the father of zoology. So, I'll mention other landmarks of missions from zoology. There are quite a lot of them, most of them laden with Nobel Prizes. Carlos Linnaeus presented taxonomic principles of animals and plants. Georges Cuvier wrote on classification and comparative studies of animal structure. Charles Darwin on the concept of organic evolution, and William Harvey on the essay on the motion of the heart and blood. Modern zoology first arose in Germany and British universities, and zoology has since changed with the growing knowledge of genetics and DNA. However, the greatest challenge to modern zoology is the extinction of animal species and habitats under the banner of biodiversity, with a timeline starting from 340 BC to 1992 AD, when Edward Wilson published the plea for biodiversity, the diversity of animal life. Zoology has become a career, vocation, and a way of life to different people, including all of us sitting down here. My research journey, trust and focus. My PhD research on the biology of 10-pounder Elaps lacerta in 3 aquatic habitats, Lekki Lagoon freshwater, Lagos Lagoon brackish water, and the coastal waters off Lagos coast show the link between the 3 habitats with the fish species in the 3 ecosystems. The juveniles of the species were found in the freshwater and brackish water, while only adults were encountered in the marine habitat. The study thus provided me with the opportunity, a unique one, an experience to master the 3 major aquatic habitats in nature, and thus predisposed and initiated my future research in fisheries, no matter the habitat. My training in 1983 and the acquisition of certificate in waste management by Environmental Protection Agency of the Department of Environment, Washington, D.C., equipped my research in environmental management and pollution studies. Thus, in most of my studies, I have preached and maintained environmental friendliness. In all my studies, I have used this opportunity to make sure environment remains as a centerpiece. It has produced my best manuscripts in reputable journals, made me keynote speakers in scientific meetings, chapters in books, amongst others I may not be able to mention here, my Vice Chancellor. Contributions to the biology of fish. The term fish, fishing, and fisheries need a brief mention here. Fish, to some, refers to the dominant aquatic vertebrates in the aquatic fauna assemblages. However, it includes other we refer to as shellfish, gastropods, bivalves, shrimp, prawns, and crabs. Fishing is one of man's most ancient food-gathering endeavors, using appropriate tools aimed at catching fish. Fisheries itself is economic terminology encompassing the state of commercially important aquatic resources, their exploitation, utilization, conservation, and management to avoid wastages. The manuscript Ugwumba in 1989 on the distribution and growth of the 10-pounder in the aquatic environment in Lagos revealed the spatial distribution and growth patterns of the species. The 10-pounder spends at least a year in the lagoons, and those in the 3rd year of life occurred mainly in the marine waters. The ecological separation of adults in the marine environment and juveniles in the lagoons was established, and this paved way for an efficient 10-pounder fishery management and exploitation. My tables 1 and 2 will show that. Ugwumba in 1991 on the food and feeding habits of 10-pounder in the freshwater, brackish water, and the marine environment shows a very successful predator that fed on the most available food item and switched to another one when the most available one was depleted. Table 3, Lekki Lagoon, Tables 4, Lagos Lagoon, Table 5, off Marine Coast. The reproductive biology of the 10-pounder published in Ugwumba and Ikusimeji 1992 confirmed the occurrence of different size groups and life history stages in the three respective environments. In the 25-month study duration, adults were only recorded in the marine environment of Lagos coast. Ripe individuals were captured all year round off Lagos coast. Figures 1 to 3 describes the gonal maturation cycles of the species. The croaker species, arguably the most popular single commercial group of species in Nigeria, was inundated with taxonomic, racial, and differential speculations. My first PhD student, Dr. Gosina Anyanwu, retired director and head of service fishery resources in Nioma, clarified the problems in 3 publications: Ugwumba and Anyanwu A, 2002 A and B, and 2003. The studies employed morphometric, meristic, and electrophoresis techniques to separate and confirm the different croaker species in Nigerian coastal waters. This paved way for an effective, sustainable exploitation and management of the resources. My second student, a Cameroonian, Professor Oben, now at the Yaoundé University, repositioned the use of hard parts in the aging of tropical fish from Lekki Lagoon. Our findings, an offshoot of our erudite and retired Professor Eso Fagade, the Grand Master, was reported in several scientific sessions across the globe. Further freshwater fish biology contribution included that of Ugwumba and Ugwumba that reported for the first time the biology of the erstwhile brackish water black-jaw tilapia in a freshwater lake Oba reservoir. Along the same line, the study reported reproduction of the tilapia species in the Oba reservoir in affirmation of a successful adaptation of a brackish water fish breeding in a completely freshwater ecosystem. Contribution to limnology. Professor L.C. Bido, Honorary Senior Research Fellow of the University of Newcastle and a Professor of Zoology in Makerere University, Uganda from 1949 to 1964, gave my most favored description of limnology. The study of inland waters, lakes, rivers, and swamps, concerned with not only discovering what these water bodies are composed of, but most especially the understanding of the complex interrelationship between the physical, chemical, and biological events that maintain them. The subject, limnology, has its main practical application in fisheries, water pollution, and public health. My contribution on limnology covered the southwest Nigeria, southeast, and the south-south. From the southwest Nigeria, Dr. Tosia Tobatele, then my PhD student, focused on aquatic environment in River Ugwumba, Ibadan, Oyo State, where he was able to establish that there were various pollution signals that included benthic invertebrates that were pollution tolerant, blue-green algae that are 92% abundant, were mostly colonial forms, all were pollution tolerant species. This confirmed the influence of organic pollution. At Tobatele, Morendikeji, and Ugwumba, 2005 and 2007. However, in Aiba Reservoir, Iwo, a tropical suburban reservoir, studies reveal reservoir water of suitable potential aquaculture features in pH, temperature, and dissolved oxygen, all within acceptable limits. Hence, feeding behaviors were very good for fish in that area. At Tobatele, Ugwumba, 2008, 2010, 2011. Contribution from Okpuka Creek, Niger Delta. My then PhD student, Dr. Bubu Davis, targeted the resultant input of anthropogenic effluents from Transamadi layout and the surrounding waterfront communities. The studies investigated species composition, diversity, distribution of plankton, epiphyton, and finfish assemblages. Further studies from that area included total coliform bacterial pollution and ribonucleic acid interference technology, a promising solution to shrimp viral diseases. Physicochemical parameters within acceptable values included water turbidity, transparency, and temperature. All of these favored algal growth. However, total dissolved solids, total organic carbon, total organic matter levels signified organic pollution from anthropogenic inputs. Other chemical parameters not within acceptable levels included chloride, calcium, magnesium, and water hardness. Nutrients, especially phosphates and ammonia, exceeded FEPA and USEPA acceptable level for natural water bodies. Pollution indicator species were very abundant, and this was the reason why the place was under pollution threats. Dr. K. Okongwu, during his PhD studies under my supervision, evaluated the water quality and ecology of the Mid-Cross River, rivers Cross, Asun, Ebonyi, lakes Eloma, and Iyeke. A rich faunal diversity in the Mid-Cross River floodplain was symbolized by about 101 phytoplankton species, 77 zooplankton species, 69 fish species. Okongwu, Wani, and Ugwumba 2009, Okongwu and Ugwumba 2009 and 2010. Of important reference was the zooplankton Daphne oblisa, a new African record. Secondly, about 10 species of rotifers were recorded for the first time in Nigeria. Among the fin fishes, 3 major intrusive species were recorded in the Mid-Cross River. Hence, the Mid-Cross River appears unique, but also complementary to the lower and upper cross, as it contains a number of endemic species and also serves as a breeding site for migratory and exotic species of the lower cross river. Okongwu and Ugwumba 2012-2013. A very rich fish trade abound in the region, with over 40% of the fish families being monospecific and endemic. Okongwu and Ugwumba 2010 and 2013. In a just concluded PhD study in the Buya River, Oyo State, Secretary, another PhD student of mine reported abundant pollution-tolerant biota of blue-green algae, copepores, and gastropods. When combined with the high metal nutrient content that were above acceptable limits, the river was classified as eutrophic and under pollution threats. Accordingly, Ugwumba in 2015 thus concluded that the water quality is deteriorating and may become unsuitable for some organisms and unsafe for humans. This shows that the so-called reserve area are not safe, hence the need for urgent management measures to protect the resources. Aquaculture. The deliberate culture in natural water or artificial enclosure containing water of economically important finfish, shellfish, aquatic plants, and other organisms for food, for recreation, ornamental, scientific, and education purposes. The history of aquaculture is still not exact. However, most of people are making guesswork about it. The general belief is that aquaculture seems to have originated in China. The numerous Chinese proverbs made the reference to the culture of fish. Prominent among them include 1, give a man fish, you are giving him food for the day. Teach him to grow fish, you are giving him food for the rest of his life. 2, where there is water, there must be fish. Hewitt in 1970 reported fish culture in Occidental Europe, which developed along with the monasteries, and this was limited to the production of food. By the 19th century, modifications, modernization of rearing methods, introduction and general use of fertilizers, and the growing use of artificial feed took off. Considerable development was involved in North America, and by the end of the Second World War, fish farm practices were forced to spread to other countries in Latin America and Africa by the following. 1, facilities offered by modern forms of transportation of fish, the constant expansion of artificial reproduction of farmed fish, and the development and use of formulated feeds. In Nigeria, records show that fish farming commenced around 1951 or thereabout. However, this was only in fish farm in Panyam, Plateau State. This has been challenged with the fish ponds by our colonial masters that was found in Ikoyi and Calabar very early before that date. Fish production through aquaculture. FAO in 1983 documented contribution to aquaculture by several countries towards fish production. In the 70s and 80s in the United States of America, values up to 11% of the overall fish production came from aquaculture. China 50%, Israel 25%. However, by 2013, China had the highest fin fish production of 60% through aquaculture. FAO 2016A. The Federal Department of Fisheries, Nigeria, 2008, recorded Nigerian aquaculture production of about 85 tons in 2007. However, FAO 2016 has put our aquaculture production to about 2,000 tons in 2010. Mr. Vice Chancellor, sir, in the early 90s, we had hinted about new frontiers from profitable for profit in aquaculture. Fagade, Dabisi, and Ugwumba in 1993, on the conservation of resources through aquaculture, advocated the culture of marine prawns, marine shrimps, as well as crocodiles for export and sales to Nigerian zoological gardens under the supervision of government agencies. The Vice Chancellor, sir, the problem that has remained the bane of tropical aquaculture has been that of inadequate fish seed and fish food. Solutions to fish seed production are found in adapting technologies for indoor production of fish seed. The success from such technologies is strongly hinged on a thorough understanding of Thalios reproductive endocrinology, as well as the underlying theories of and the techniques of induced spawning in fish. Thalios reproductive endocrinology. The nervous and endocrine systems of Thalios fishes, as in other vertebrates, act in concert to coordinate reproductive events. The reception of environmental stimuli, temperature, amount of rainfall, and daylight are mediated by the nervous system, and this involves the message or the passage of information from sensory receptors to the brain. This neural information on reaching the hypothalamus determines the activities of the pituitary gland by way of chemical messengers termed gonadotropin-releasing hormones. These, in turn, will stimulate the pituitary to release into the general circulation a hormone whose target is the gonad. This hormone is termed gonadotropin. Techniques in induced breeding. There are two approaches to induction of spawning in fish: hormonal and environmental approach. After treatment, the fish in each approach may be stripped for artificial fertilization or left for natural spawning. The fertilized eggs are then incubated, and the hatched larvae are raised to fry or fingerling stage in the hatchery, which may be indoor or outdoor. Figure 4 shows the hormonal and environmental approach. Figure 5 shows the schematic diagram of endocrine control of final oocyte in fish for females. In females, the hormonal treatment of fish is known to achieve the following. It stimulates the release of gonadotropin-releasing hormone, which causes the pituitary to secrete gonadotropin. When a certain level of gonadotropin is reached, the germinal vesicle migrates to the periphery and the theca, and this will cause the release of another steroid, maturation hormone. This one is known to induce the general vesicle breakdown, simply means resumption of meiosis. In males, this mediation of gonadotropin action on the testes is to bring about spermiation, and seminal hydration is still not very clear. Our contribution to aquaculture. The paper Ugwumba and Ugwumba examine aquaculture options and the future of fish supply in Nigeria. This has been due because of the decline in returns from catch of fisheries, thus using aquaculture to bridge the widening gap from domestic demand and supply of fish. The review recorded species which have shown through studies ability to tolerate culture conditions in terms of feeding and seed production and acceptability of artificial feed. I've made in zoological drawings 15 other species that we can use to augment our culture of purely catfish and tilapias. I've also included ornamental fishes for local fishermen to adapt in culturing fishes. The same paper reviewed several documented reports on artificial propagation, including when we use overprim in the limpe option to spawn fish. Limpe option. The synthetic mammalian luteinizing hormone releasing hormone and its analog and a combination of dopamine analog have been found promising in the continuous search for better and effective artificial breeding techniques. This combination, simply known as overprim, is the most viable option to avoid the production of low fertile eggs. Ugwumba, Soumi, Bubu, and Ofuan in 1983 studied overprint efficacy in artificial spawning of Flaria garriapinus. Conclusions from our findings include the following. When time to peak breeding period, a single knockout dose of overprint was enough to induce ovulation. Maximum latency period of 16 hours was overprint dependent. Very high number of fertilized and hatchable eggs, as well as high larval production, can be obtained with the use of overprint. High larval production indicates overall good egg quality, effectiveness of overprint in inducing ovulation and spawning in the African catfish. This method is a further confirmation that the manipulation of the hypothalamus pituitary gonadal axis with the aid of synthetic hormones and non-hormonal agents can produce all-round year egg for fish farms in Nigeria. The advantages of limp action include effectiveness of labor, reduced stress of broodstock as fish is handled once to give a single knockout dose. This is not difficult to apply. Furthermore, overprint is stable with precise dosage and is not immunogenic. However, it should be noted that overprint is imported into Nigeria and is thereby expensive. Despite all this, it is still in high demand because of its effectiveness. In addition, this method has shown that fish farmers could provide simple workable experimental sites where fishes can be produced in the farm to reduce stress due to transportation, as well as avoid being supplied with trash fish by dishonest fry vendors. Hyperfixation along this line, despite its apparent sophistication, remains a method of choice for rural fish farmers, since it may be carried out in the absence of such amenities as centrifuge, electrical balance, and refrigeration. My Vice Chancellor, sir, our other studies examine the best options for food and feed of fishes in captivity to a level that it won a national award by the NUC for Best Doctoral Thesis in Biological Sciences in 2007. From the same fisheries unit in this department. I don't think that clap was for me because this story will be told at the appropriate time by the appropriate person. Environmental research. Aquatic toxicology. Joint research on aquatic toxicology has been ongoing in ecology and environmental biology research unit of our department. This unit has been a melting point where staff from other units join order to conduct research, bringing all aspects of zoology to bear on ecology. After all, sir, we're all ecologists, seeing the animals and their world from different perspectives. These are my few humble contributions from this lab. Okori and Ugwumba and Oko in 1992, Okori and Ugwumba and Obafemi in 1993 conducted static bioassay on effects of Piper guinense on the fingerlings of Oreochromys nauticus. The study showed that ethanolic extract had pesticidal potential, and we advocated the possible use of this plant to selectively eliminate unwanted trash fish in fish culture. In a Senewa and Ugwumba 2010, we reported the growth response of the catfish exposed to water-soluble fractions of detergent and diesel. Generally, detergents negatively affect the growth and survival of the catfish more than crude oil. Hence, in Nkpodion, Ugwumba, and Senewa 2016, we employed the detergent to check the enzymatic and protein activities of the same fish. The studies show that the detergent induced toxic effects in form of enzymatic degradation, organ damage that can make the organism vulnerable to diseases and eventually die. Olarimoye, Bakare, and Ugwumba 2015. My Vice Chancellor, sir. Bakare was made a professor yesterday, sir. Thank you, sir. On the pharmaceuticals as environmental contaminants of concern in Nigeria, against the background that very little information exists on the presence of pharmaceutical active compounds in faunal and floral matrices, very little information from the African Regional Group, and virtually none from Nigeria. Our ongoing research currently employed recent advances in analytical protocols to detect and quantify several pharmaceutical active compounds. In a targeted snapshot reconnaissance investigation of surface water and benthic samples from 6 locations in Lagos, Nigeria, we covered a range of pharmaceuticals, including antibiotics, estrogens, non-pharmaceuticals, including anti-inflammatory drugs and lipid-lowering drugs. Screening showed that out of the 37 detected, about 4 of them exceeded allowable concentration. The pharmaceutical active compounds present established by this investigation, especially in a very available and misused pharmaceutical families, showed the probable effects of unregulated use, their disposal, and sewage impaction in Nigeria. The study suggests the need for Nigeria not just to acknowledge such issues as an environmental concern, but as a priority for quick action as in the developed world. In further studies along the same line with our German counterparts from the German Environmental Protection Agency, Olarimoye, Bakari Ugwumba, and Hen in 2016 looked at industrial, domestic, and hospital sludge from Lagos, Nigeria. Diclofenac mostly present in all samples at a very high concentration, 2 times more than what it is reported in sludge samples worldwide. This suggests that there should be a comprehensive water monitoring campaigns in high-density areas in Lagos. There's need to investigate the groundwater, tap water, drinking water, manure, soil, and sediments as additional matrices of concern to be able to generate a holistic picture of the spatial environmental presence and concentration of pharmaceutical active compounds. My Vice Chancellor, sir, in Adiogu and others, 2016, we reported endocrine disruptor molecular responses occurrence of intersex in our reservoir in Ibadan, University of Ibadan. From studies in our counterpart laboratory in Norway, we found out that we observed endocrine disruptor responses associated with contaminated concentration included intersex, elevated expression of vitellogenin, zonal radiator protein. All these are negative sensitive early warning signals of disruption in reproductive processes. This suggests that the contaminants elicited severe endocrine disruptive effects in Oba Reservoir Bauta, an important source of water for University of Ibadan as well as fish. The Vice Chancellor, sir, the health consequences is probably the biggest issue here regarding contamination burden in our dam. The study furthermore contributes immensely to the establishment of biomarker responses or monitoring protocols for feral fish species and human health in Nigerian rivers and streams, providing useful information for suitable management of environmental and potential human health. The figures 9, 10, and 11 will show normal testes and ovary, and another one mixed with ovary and testes called intersex. The other ones are the histological expression of that formation. Contribution through books, chapters in books, reports, and manuals. The Vice Chancellor, sir, over the years, I've been involved in teaching and research, as well as books, writing manuals and reports for the industry, research, non-tertiary and tertiary institutions. Motivation to write secondary school textbooks came from my experience from the years I served as examiner for senior secondary school and West African examination boards. The marks obtained in biology were comparatively low in the 80s. To me and my colleagues in the department, one of the problems was how students answer biology questions. Also, students scored low in questions on conservation and biodiversity, as well as wildlife and ecology at the ordinary level examination. In Odaibo and Ugwumba 2003, Mastering Series, Macmillan Press, Nigeria Limited, we included model questions and answers in all chapters of the book with a good coverage of the biology syllabus. This book received commendation even from our undergraduate students that found it still relevant and appropriate at the university level. In a follow-up book, I was invited to join secondary school teachers to write. We adopted the same pattern in Adebayo Begu, Ugwumba, Salam, and Sarumi in integrated science for junior secondary school. Along my research area, I joined to co-author a fishery research textbook Ugwumba and Ugwumba 2007, Food and Feeding Ecology of Fishes in Nigeria. As director of Zoological Garden, when we embarked on the secondary school children's project, I joined in writing the following: Okwilavi and Ugwumba, Andy and Sally Zoo Surrey, University of Ibadan, Ugwumba 1995A, the Zoological Garden, University of Ibadan, Reminiscences and Future Survival, Ugwumba 1995B, Apes in the Zoological Garden, University of Ibadan. For the international audience, I joined in writing the following: Fagade, Ugwumba and Ezenwa 1992, Introduction to Aquaculture, Fagade and Ugwumba 1992, Species Selection, Training Program Reference Manual for Aquaculture Extensionists in Commonwealth. For the Nigerian Association of Aquatic Sciences, I joined to edit Sustainable Utilization of Aquaculture in Wetlands. For the Big Builders Education Africa, we wrote a chapter, Ugwumba and Ugwumba, Poverty Alleviation through Aquaculture. For the NGOs, sir, the following are my humble contribution. From 1993 through 1997, 98, 99, to 2001 and 2009, I was able to write fauna studies, hydrobiology, fisheries, and so on for different projects for Total, NMPC, and Chevron. All this culminated in my being invited to write a lead paper in 2009A, Marine Resources of Nigeria, Need for Conservation. I was also invited to present a lead paper, Current Status of Environmental Pollution in Nigeria, Water Pollution. And finally, I joined my colleagues in Southampton University on to write the Ocean Biodiversity Survey Report, which I should mention in a short while. My Vice Chancellor, sir, in all this, I was called different names: hydrobiologist, scientist, animal scientist, pollution consultant, marine biologist, limnologist. However, sir, I am simply a zoologist, seeking, studying, conducting research in new areas to impact my students and the society. The Vice Chancellor, sir, my second research activities in the marine environment was now that of an environmental evangelist. In doing so, I discussed an environment as comprised of animate and inanimate influence which bears on man, his life, health, and livelihood. During my earlier research activities, I used fish in the study of fisheries of the lagoons and coastal waters. Hence, quite many of them were sacrificed, however, for a just cause, to improve, maintain, sustain, conserve, and perpetuate the species. Earlier studies in this zone are quite many, but I would like to mention the one of late Professor Emmanuel Obot, a book titled Oil Rivers and the Armpit of Africa, where he described the area as a biological hotspot. The Niger Delta is a repository of fish biodiversity with a minimum of about 314 species from 158 genera and 64 families. Professor Martin Ogbe submitted wetlands, water, and poverty eradication. He also mentioned some of the problems in our wetlands, and some of them we take for granted. In my own submissions, Ugwumba 2009 and 2011, the current status of environmental pollution in Nigeria, 1, marine resources of Nigeria, some yet unknown need for conservation, I listed the problems of the coastal zones and the way forward in pollution studies and abatement. Wetland services we take for granted include water storage, flood water, carbon storage, climate change, and transport. However, major wetland values include fish, finfish, shellfishes, fruits, fodder, fuel food, medicinal plants, timber, other building materials, as well as reeds and palm. In Ugwumba 2009, I listed the problems of the coastal zones to include overpopulation, environmental pollution, gas flaring, oil spill, sewage, solid waste, climate change, sea level rise, coastal erosion. I will now describe plates 1 to 12. Plate 1. This is the Niger Delta before pollution, followed by the thick rainforest. This is the green nature, protecting, incubating, preventing all our resources, including our blue gold, the oil. I listed hundreds of thousands of species that can come from this zone. The next figure, we began to look for oil, began to excavate, to dig. The more oil we get, the more we dig. The next slide shows the impact of this oil exploration, degradation. We cannot grow food anymore in that area. Next slide, the tankers are falling, throwing all the oil into the sea. Fishes are disappearing. Next slide, the simple young boys think they are picnicking. They don't know what comes from environmental pollution of what they are watching. Next slide, sir. Oil spill fire blowing everywhere. An outburst of oil pipe, either intentionally or by accident. This is followed by this picture, which I describe as the face of poverty. Followed by the sources of water. Most of this water are not even good enough for our domestic animals. From the information from our medics came some very sordid pictures. Malnutrition because of no harvest and several associated congenital malformation as a result of all this. My Vice Chancellor, sir, I listed marine and brackish water finch fish species, commercial important species, marine mammals. In doing so, I was trying to point towards the fact that with these huge amount of resources in our coastal waters, the need and rationale for conservation becomes an urgent issue for research and government intervention through sustainable management and preservation. Rationale for conservation of the coastal zone. Conservation is defined as the highest sustainable quality for mankind by the rational utilization of the environment and the resources therein. Resource is defined as a commodity ready for use or available as needed, as at when new, because it is renewable or non-renewable. Most clearly recognizable resources consist of living organisms characterized by being able to reproduce and regenerate their population as long as environmental conditions remain favorable. Conservation of species enables every species to fully develop its global values of biodiversity, specifically, ecologically, scientifically, and educationally. Aquatic resources are assumed to be inexhaustible. Hence, the general belief, just go and collect. This is very wrong because ruthless exploitation of habitat will cause habitat loss, desecration of the habitat by oil industry, all this with hastened drastic reduction and extinction of species. Further reasons in support of protection and conservation include the fact that there are many resources still unexploited, some of them yet unknown, some of them their values yet untapped. In my submission on the World Ocean Day 2011, I attempted to show resources still in different museums, awaiting full identification from our studies from the ocean as well as from our counterparts in the Serpent group. Challenges that face these studies which slow down capture and identification include remote environment, limited existing knowledge, evolving impact assessment methods, need for new information, pollution monitoring, pollution control, abatement, prevention, and way forward. These are the emerging trends on above. The first one is already on ground, environmental impact assessment. By law, all new facilities or existing ones are supposed to conduct EIA to be verified and clarified by appropriate agencies. Second one is Environmental Sensitive Index. This is an initiative from RECTAS, Regional Center for Training and Aero-Based Survey, OAU Leife, use of remotely based geospatial data using appropriate hardware and software to depict variability in water. There is satellite imagery, which is a Norwegian initiative, use of satellite imagery based on density and color of water to be able to look at coastline communities and assess contamination. This initiative is said to be very expensive. International Atomic Energy Agency, a proposal for partnership. This project is resident in Ministry of Environment, Abuja, and the operational base is, and national coordinator is in Department of Animal and Environmental Biology, University of Port Harcourt. Their mandate is to undergo baseline studies of hotspots for identification of life forms, as well as show resource degradation, renewable resources, and depletion. The question for this initiative is that nuclear material emission, incidents, accidents, and nuclear contamination, are these safe? My Vice Chancellor, sir, I got invited and attached to Project Serpent, scientific and environmental ROV partnership using existing industrial technology. This is the opportunistic use of remotely operated vehicles to gather environmental data through video cameras, live pictures, and different physicochemical parameter equipments. In this study, the benefits include that the equipment is already in existence. There are about 220 ROVs in the world, 200 of them in the oil industry, about 20 are found in the universities of the first world. The important is that you can identify the organism in the field. You can also identify through deep sea videos the organizational ecosystem, habitat interaction of these organisms. The Serpent team is home-based, is the National Oceanography Center, University of Southampton, and their representative is Dr. Daniel Jones. The contact and the link and sponsor is Total Nigeria, with their representative, Mr. Charles Maibure, a graduate student of this university. Country scientists include my friend, Dr. Shina Adegbaye, geophysicist from Nigerian Institute of Oceanography and Marine Research, and my humble self from this university as the marine biology, pollution, and environmental consultant. The picture of our ocean trip I will now briefly go through with you. Plate 13 showed the biodiversity hotspots of the world. If you check in Africa, towards the armpit of Africa, there's a biodiversity hotspot. We are looking at the armpit of Africa, the Niger Delta. After a 2-hour trip into the deep sea, we were thrown in what is referred to as a basket throw from the oil rig in Ulsan into the research vessel. The next slide is where we are landing from 75 meters to the floor of the vessel. This, plate 16, is a remotely operated vehicle. Simply, it's a robotic science that has appendages, hands, manipulators that they use in the oil industry to close the oil well in the deep sea bed when they don't want to pump oil, and it can open the deep sea bed oil tap for oil to flow. It is also used in maintaining the deep sea bed oil floor so that it will not be eroded. So what we'll do in this research is to use any window of opportunity when this robot is not doing its natural work to attach our equipment for our studies. Next slide. This is our equipment. The one we attach on the body is the planting net attached to the body of the ROV. This is the ROV being sent into the deep sea bed. For this research, we were about 900 meters deep in the water. This is the power operational room where we can now monitor, move, direct the ROV to do whatever we want it to do. It can collect samples, it can dig mud sample from the floor, it can set a trap, a basket trap for fish, it can do whatever it wanted to do, remotely controlled from the vessel. All the equipments we used for this operation were fabricated in the lab by us. This is the research team. From left is the representative from Total, my humble self wearing the UI flag, Dr. Achino, and Daniel Jones from Southampton. This is the research team with the chief, the captain of the boat, and the chief boatman. Fishes, some yet unknown. Against the background that our planet is largely composed of water with depths reaching up to 4,000 meters in some areas, little is known of the creatures at these depths, as well as the nature of the ecosystem supporting them. As we turn to the deep seas for a wide range of solutions to mankind, now and in the future, wise use of these resources in the deep sea bed demands knowledge of these resources and the fragile environment they reside in. Fishes that abide in this deep sea abyss have displayed a variety of adaptations, modifications, behavioral responses to survive and live in the often dark ecosystem. Adaptations range from torpedo-shaped, bioluminescence, balloon-shaped formation, attachment surfaces to escape from the predators as well as from natural water movement. Fishes, the masters of the aqua world. Sir, I will present now information from our Ocean trip and our partners from the Oceanographic Center, fishes that are yet unknown and yet unidentified. Plate 23, the piglet squid. This is usually bloated in an excited state. This is the flagship appearance of a squid when the ROV lands at the bottom of the sea. It's a totally dark environment, but when the ROV lands, the piglet squid swallows water and looks like a piglet to scare away any predator. Secondly is the unknown theropod gastropod, followed by an unidentified sea anemone, followed by another unidentified sea lily, followed by another unidentified comb jelly, followed by another unidentified king crab, and we were able to identify Antimorosa rostrata, the beautiful black pearl of the deep sea bed, and unidentified monkfish, a very camouflaging predator, followed by an unidentified greyfish and unidentified ray and skate. Concluding remarks and recommendations. The Vice Chancellor, sir, over the years in our pursuit of my academic career, we have joined in formulating policies. We have joined to make statements, bogus statements that have not solved any problem, because we have not resolved the pertinent issues before we advance to the next robust and complex issues. My Vice Chancellor, over the years, my professional colleagues have echoed the promise in every decade, Fish for All, 2005, 2010, 2015. In fact, now we are saying Fish for All, 2020. If we have not done so in the last decades, it is the next few years that we'll be able to make fish available in correct quantity and right price. However, I will confirm, in line with other honest fishermen, by the following. We should undergo a vigorous promotion of fisheries as a viable commercial venture to be modified by the fisheries sector. We should continue to train and retrain at different levels of various aspects of fish culture, prevention, and marketing. We should ensure that all stakeholders are all registered members of Fisheries Society of Nigeria to coordinate best practices and dissemination of our findings and policies. We must provide fisheries statistical data that is verifiable, effective fishery legislation, resolve fishery conflicts, and reappraise and eliminate factors limiting abundance of fish. We must further give way to aquaculture and restocking, rejuvenation of depleted water bodies, production of fish seed and fries, comprehensive stock assessment, providing fish inputs, and essential linkages. Sir, for all this to be done, we must consider the health of the environment first. Our inland waters have become sources of livelihood for many. Hence, they should not be turned into sinks of impurities. It is time for the appropriate agencies to get on with their duties of monitoring, regulating, and controlling what gets into the water bodies. My Vice Chancellor, sir, the National University Commission present benchmark for academic programs need to fall in line with present realities. Once again, the University of Ibadan has approved new academic curricula submitted by different departments. I will speak and defend what we did in biological sciences of the Faculty of Science. We have taken botany, microbiology, and zoology to an applied science to the next decade. This is a wake-up call for NUC to adapt the University of Ibadan style, because it is the first and still the best. My Vice Chancellor, sir, we must appraise the water bodies within the campus and enhance the quality of what is entering from Ibadan metropolis as well as within the campus. This must be monitored and checked before use by consumers on campus. Acknowledgements. To God be the glory for fulfilled dreams. I owe it all to my father, who passed through the University of Ibadan on his way from Furoba College, Sierra Leone, where he bagged inter-BA honors European history. He vowed to have a son pass through this university. Out of 4 of us, I was the person sent here. Here am I, son of Mr. Nelson Ugwumba, THOT, headmaster principal in 11 primary schools and 6 secondary schools in Cross River, Aqua, Imo, and Abia states. May his soul rest in perfect peace after 91 years sojourn on earth. I salute all my teachers, for the profession was a household name throughout my teenage life. My sincere gratitude goes to the first generation of Ibadan School of Zoology, from the transitional mother of all, Dr. M.B. Hills, to late Professors F.M. Okoli and Professor S. Otoye, Professor M. Wagu, W.W.D. Mother, the Grand Master, Professor S. Fagade. From the Aqua School of Zoology, our school there too. I salute Professor C.I.O. Olaninyo, product of Ibadan, Professor K. Kusemeju, and Professor Willie Odiete. My other teachers, Professor Tubanjo, who is the head of the department, is seated right here. You're welcome, ma'am. I appreciate all my students who I taught at all levels, and I thank all my colleagues in the Department of Zoology. I thank you all because we have continued to follow the academic foundation of our relationship handed down to us by our predecessors. Sometimes they were fearful. They handed us the culture that is devoid of geographical affiliations. I'm grateful to the Faculty of Science family, the elders, Professor Gumola, Okories, Fanero, Okogus, Adelekes, Odiaka, Adesemoju, Osho, Hussein, just to mention a few. To my colleagues and friends, Professor Daibo, Oniawa, Vice Chancellor Sir, Onilude, Fagade, Adebowole, Oyelaro, Aleru, Folorunsho, just to mention a few. I was supervised by the best. Professor S. Otoye, BSc Zoology, the author of the African millipede Afolobina Yoruba, Professor Eso Fagade, the inventor of the use of hard parts in aging tropical fisheries, the Grand Master, sir. Professor Kola Kusimeju, the doyen of interrelationships in the aquatic perturbations. I wish you were here, sir. I know you say you won't come. I pray for your families, sirs, that they will receive the same unparalleled guidance like I got from you. To my professional colleagues of the Fishing Society of Nigeria, hereby fully represented by the Chairman of President Committee of Fellows, I salute you all. From the Zoological Society of Nigeria, which I'm currently the president, I repeat my promise to you, I will serve you with all I have. To my in-laws of the Krenamitima Beekpo of Eyabasi Oro Nation, little will you know that you are dealing with princes and princesses until you are told. May the Lord God continue to reward you for the joint family you have forged between my family in Aruchuku through marriage. From the same family came the finest policeman Nigeria ever groomed, late DCP Osun Ekbo, my friend for life, crime buster par excellence, rest in peace. I bow to the teachers of our nation, from our patriarch, late Alvan Ikoku, late Tichudo, and others. They all were the mentors who we all lived, that all we saw in this life was teacher as a career. I salute the president and executive committee of the club, senior staff club, and members. Indeed, the ability to supervise and manage the huge investment by this university in the staff club is commendable. History will always place you amongst others. I'm very grateful to the National University Commission for exposing me to academic standards and ethical issues through training, workshop, and accreditation exercises. These strengthen my vision on academic issues, especially on the instrument of academic standardization. I thank the Vice Chancellor of Ibadan University, Professor Reverend E. Osage JP, the Registrar, Mr. Idiokoro, the Librarian, Mr. Yakubu Izebakai, who provided me with the serene atmosphere to think and write this lecture. I thank the University of Ibadan for, apart from my basic academic assignment, allowing me to serve in different areas, and all these have improved me for life. Finally, I thank my best and special friends. My daughter, the town planner we call her, Bella Uchechi Ugwumba, who currently completed her master's from this university. My son, Dr. Alex Obinna Ugwumba, a private medical practitioner. My sister-in-law that took care of my children when we were running around looking for research. Dr. Jiriadin Umar Ekbo Fashional, Senior Lecturer, Ignatius University, Port Harcourt. To my wife, Professor Bia A. Ugwumba, FFS, Head of Department of Zoology. We have come a long way through God's grace. Given another chance, I will marry you again and again and again. In you all I live, and I have no regrets. Fish for all, and for now. Could God be the glory. Thank you all.